Hi, everyone. I'm Ben Barad. This is my second time participating in this course, and it was really fun last year. I ended up I gave sort of a short talk, and then at the time we had just gotten really excited about doing filament segmentation. So I actually went through doing that um, live, which was maybe a bold move. Last time it went pretty well, and it was a lot of fun to sort of do that. The video of that's online, so I'm going to give a talk today. And the segmentation workflow for filaments hasn't changed that much, so I will probably not do that again, but the video is available. But what has changed a lot in the last year is that segmenting membranes has become more or less trivial now, and it was quite challenging before. So I'm going to show that off, and you're going to get to see like a full tomogram get segmented, hopefully in about 10 minutes at the end. And that's like, the a little bit of movie magic. But first I wanted to talk about, not how we're going to do it, but like what segmentation is and why it matters. And my lab at OHSU is really centered around the ways that we can creatively use segmentation to advance cell biology with tomography. So you may have already seen this tomogram. I would say that this is probably the most famous tomogram, or cellular cryo-electron tomogram, at least. So this was a beautiful Golgi segmentation that was segmented by Ben Engel at the time, and it was this. This is a from this grayscale image on the left where you see all of this stuff, and we as humans are quite good at finding it. You're seeing a single slice. With a with a segmentation, suddenly you get to build a three-dimensional color map of what you're looking at inside of the cell. And so, we definitely joke that this is the segmentation that sold 100 Aquilises. This is very much like this paper was very motivating for people to start milling. And segmentation is really a starting point for many different kinds of analysis in cryo-electron tomography. So I'm going to talk about sort of those first, and then we'll talk about the how. So I tend to be a little bit sloppy with my language some of the time, but I thought it would be worthwhile to take a moment and talk about the terms that I'm going to use very loosely. And like, if there's other terms I bring up, please feel free to pop something in the chat, and like I can pause and describe what I'm saying, because I don't want people to feel lost by jargon. And the field just has a lot of jargon. So segmentation is and specifically it's called a semantic segmentation, is a map. It's the same dimensions and number of voxels as your tomogram. But instead of having grayscale values, you have a series of numbers from 1 to whatever that correspond to the classes of each pixel. So you might say these pixels belong to membrane, or these pixels belong to ribosome, and so on. So it's a very specific kind of data structure. I tend to think of the segmentation process much more generally, and I think about geometric models, where it's sort of all of these different possible ways of encoding ultrastructural features in sort of compute-friendly ways. So, segmentation kind of is one. It doesn't really encode geometry, but it has information. I would also argue that like particle locations and orientations in a star file are often considered as a geometric model, because people work in tomography, when you're doing sort of visual proteomics, this molecular sociology idea, you use those particle locations and co-orientations to learn a lot about how proteins interact with each other. And I might be interchangeable between voxel and pixel, but a voxel is like the three-dimensional equivalent of a pixel for all intents and purposes. So with that out of the way, you guys have gotten an introduction to cryo-electron tomography, but I thought I would sort of talk about why I am excited about cryo-electron tomography in particular. And that is the ability to see sort of both the molecular structure and localization of proteins at the same time as you see how they're impacting cellular architecture. So this tomogram is a tomogram showing a mitochondrial fission site. So mitochondria undergo fission and fusion. And in my postdoctoral work, my sort of job was trying to investigate mechanisms of fission. And you get a beautiful picture like this that, I think is immediately very attractive. You can see the mitochondria on this. So mitochondria have two membranes, so they're very easy to tell which one is a mitochondria. It's this thing coming sort of down through, it's moving out of this Z plane and up and around here, and then back down and up this way. This is endoplasmic reticulum crossing over it. So endoplasmic reticulum marks sites of mitochondrial fission. You can see actin filaments. You can see in the mitochondrial cristae, which are these invaginations, that is where all of the energy in the cell is generated. They're like the capacitor of the cell, essentially. These are ATP synthase, these little lollipops hanging off the side of the cristae. You can see ribosomes coming on and off membranes. And if you look really closely, you can even see different proteins that are on the mitochondrial inner membrane that are causing local membrane deformation. And so, this is really exciting and powerful as a technique. The challenge is that all of these things are kind of happening in a mishmash together, and you have to like pull out the things that matter from this soup of data within the cell. We don't have this molecular specificity of like fluorescent imaging. Even with like CLEM, you only get like partial localization. So, within cellular tomography, you can really do a lot of different things, and they range from sort of cell biology questions to subtomogram averaging. And so, you can do quantitative ultrastructure, which is what I did a lot of in my postdoc. You can ask questions about visual proteomics. You can talk about subtomogram averaging, where you're recovering like detailed molecular information about protein structure in the cell. And I think that, the grand promise, the reason that many of us that are in the field got into tomography is not to do one of these things, but to do all three in a unified way, and to understand how protein structure and localization impacts ultrastructure and vice versa. And to sort of do this scale crossing into the mesoscale sort of approach. And this has been really challenging for a variety of computational reasons. And the research that I do is really centered around this hypothesis that using segmentation as a starting point, understanding the cellular part, you can gain a lot of power to do sort of, a top-down approach to then you doing visual proteomics and subtomogram averaging in addition to ultrastructure, all using segmentation as sort of a leaping off point. So yeah, why segmentation? Is because it enables all of these really cool things. So I showed you this beautiful picture of the Golgi network, but we're basically gonna start, it's a hypothesis generating tool. You can see stuff in 3 dimensions. But I guess what I would like to sort of emphasize is that doing a segmentation is more than just a pretty picture. That you can extract a lot of quantitative information about membrane ultrastructure and other sort of features using segmentation as a guide. So that was sort of the main thrust of my postdoctoral work, is that we really got interested in understanding some features of mitochondrial organization, and ended up building a whole toolbox that you can now use to use membrane segmentations for a variety of different things. So a little bit of like science background. We were studying mitochondrial remodeling during endoplasmic reticulum stress. So this is a drug that drives PERK stress. It's one of the arms of the unfolded protein response. And our collaborating lab had discovered that mitochondria undergo a process called stress-induced mitochondrial hyperfusion in response to ER stress. And we knew that this was sort of protective against cell death, but we had a lot of questions about how was the mitochondria getting elongated? What are the features that are changing in the mitochondria? Why are they doing this? Sort of what are the functional roles? And one of the really cool things about mitochondria is that they're, one of the classic examples of form follows function. And so they have these very complex shapes, but they're very directly tied into different aspects of function. And so this is a really exciting sample for us to look at by tomography. And we just wanted to know, yeah, how does ER stress alter mitochondrial membrane ultrastructure? So we do what's called baby clem, or I like to call it that in the lab. So we're not doing careful localization. We're actually using fluorescence imaging to understand the sort of functional state of the cell for each cell that we're imaging. And we can prepare thin lamellae within those cells. Um, and then we can take tomograms. And I think this is really, for us, these were the pair of images that launched, sort of my entire scientific path, but I think still is very like impactful to me today. So on the left is what our normal mouse embryonic fibroblasts look like under the beam. So you can see hopefully you can see the mitochondria in the center here. You can see these little circles. These are mitochondrial cristae. So they look like circles because in 3 dimensions, they're sort of tubes that are moving in and out of this Z plane. So we call this a tubular cristae architecture. And this was sort of the norm for what we had seen in these cells. But as soon as we treated with thapsigargan, this ER stress drug, the cristae instead formed these sheet-like patterns. So these are discs that are stacked up next to each other. And this is exciting for a few reasons. We had like a very clear drug responsive change. We do know that in some kinds of cells, particularly cells with more energy expenditure, these cristae architectures are actually the default for the healthy cell. So we don't think we're capturing some like crazy off-target thing. We think we're seeing the very normal cellular remodeling that you would expect to see under sort of a range of conditions. And this really gave us sort of a lot of exciting potential for understanding the mechanism of what the value of this hyperfusion phenotype was. There's a downside though, which is, I don't know how many of you are cell biologists or even if you read cell biology papers, but no 2 cells are alike. No 2 mitochondria are alike. They're each their own special snowflake. And so taking one picture of a mitochondrion does not teach you mechanism. And so really what we need is to be able to take lots of pictures of mitochondria. And when you do that, you can then begin to see patterns. But the problem then becomes, well, how do you like convert these things that you see into, quantifiable observations? Most importantly, quantifiable observations that you can like be like, yes, this is a statistically significant change and not just sort of the random distribution of mitochondria we happened to sample. So this comes down to segmentation. So segmentation, instead of just being in grayscale space, we can now begin to think in 3-dimensional space. So I was using a tool that I'm going to talk about more at the end, but this is a tool called TomoSegment-TV, which until a year ago was the tool that we used full-time in the lab to do semi-automated membrane segmentation. And I say semi-automated because it'll segment all your membranes, but then you have to go into another tool that I'm going to show you today and tell it which membranes are which. So like, I colored certain membranes pink, certain membranes purple here for the inner and outer membrane, and then the ER is blue. But so now we've gone from being able to see in 2D to being able to see the full 3D structures. But we're still not able to very easily do quantifications with this kind of data. And the reason for that is, going back to the segmentation versus geometrical model thing. And whenever I talk about segmentation now, I'm like, we shouldn't be just doing segmentation. We should be doing modeling. But this is very much akin to the difference between Minecraft versus, Mario. When you're looking at the way that these games work, you have a pile of voxels in the Minecraft world. And those, each block is its own thing. And they don't encode any kind of geometry relative to each other. They rely on your eyes and your brain to kind of like make geometry work. Versus polygon meshes like this Mario polygon have direct encoding of orientation and connectivity of the different triangles in them. And so if we want to be able to ask questions about geometry and organization, we need to build high quality polygon meshes. And when I started working on this, I thought that would be very easy because there's this entire world of computational geometry that had been doing this kind of thing for years and years. But it turns out that because of the limitations of electron tomography data, particularly because of the missing wedge, these tools generally don't work very well. So here are two examples of different ways you can do it. One is this earlier Marching Cubes algorithm, where you can see that the membrane gets really like shredded in the process. And alternatively, you could do what structural biologists do with their maps, which is to make an isosurface, to just like make a mesh that wraps around your structure. And that seems great. You get beautiful surfaces, but then when you try to quantify curvature, for instance, which you can see here, the tops and the bottoms get totally high curvature, like blow everything else out, because you're just artificially wrapping around a fake edge of the membrane because you just don't see the end of it. And so neither of these was really good enough. We ended up doing our own thing, and we took advantage of self-driving car technology, basically. So there's researchers at Microsoft in the mid 2010s were thinking about, well, LiDAR from self-driving car generates these point clouds, but you never see a whole object. You see like a little face of the object. And that's actually very similar to the kinds of pathologies we have in cryo-ET. So I was able to find a tool called the screened Poisson algorithm from this field, and adapt it to work with tomography data. So you take your segmentation and you make it look like that self-driving car data. You convert it to an unoriented point cloud, and then you kind of guess what the normals are using a neighbor-based algorithm. And then you can do this transformation into the surface mesh. So just to show you, this probably looks just like the segmentation, but it's actually a series of little points. And then we calculate these normal vectors. So these are saying, like, for each one, like, what's the direction that points me away from all of my neighbors the most, essentially. And this is really the critical step, is estimating the orientation of these points. And then you can reconstruct and mask out a mesh with the screen Poisson algorithm and get these structures. So this is a mitochondrial outer membrane. It's really long and sort of simple. You can just sort of do it. But it also works for like arbitrarily complex membranes. And so for the first time, we're really able to go from semi-automated segmentation all the way through to these meshes now where we have geometry encoded in a single fully automated step. And this is now, these are scripts that you can download and run. This step takes about 1.5 minutes per tomogram typically. I did want to note that as good as this step is, it requires some parameters. And so I know that I get emails from users about, maybe once a week on average, asking me for like why their meshes are not coming out well. And often it's like, well, there are these parameters you can try playing with. And this is a problem, because what we'd like is for this to be anyone can sit down and run this software. And there ends up needing to be just that little bit of expertise that I think is limiting for getting people to give it a try. So I had an opportunity to collaborate with a group earlier this year, and they had this idea, and I was incredibly impressed. In basically like a month, they were able to come up with a better algorithm that now is completely parameter free, and makes smoother, better surfaces. And so sort of in the very near future, we're adding this to our workflow. We've been testing it already, and hopefully that means that sort of going forward, you'll need a lot less prior knowledge to start using our tools. But anyway, once you have a mesh, what do you do with it? So we did all of this work to learn about connectivity and geometry. So once we do that, we should make questions, answer questions more generally about connectivity and geometry. And so you can make, what I would describe as the very basic toolbox of ultrastructural measurement. So we can measure distances really accurately. Like we can look at the distance between two different membranes. On the left here, you can see the distance between the inner and the outer membrane of the mitochondria. In yellow is really closely oppressed membrane, and in blue, they're further away. And what's happening here is that those blue spots are marking mitochondrial cristae junctions. Because if you have like your flat membrane here, but the junction folds in here. This bit of membrane in the middle doesn't have anything right opposite it to be closer, and so it gets further away. And this was really exciting because, like, we spent a lot of time marking junctions by hand in the lab previously, and this gives us a really nice sort of starting point to begin to do automated selection of junctions. We ended up doing it in a slightly different way. I'm going to talk about that a little bit later. You can measure curvature. This is what I'm most excited about. So here in blue is like really flat, in red is really curved in the membrane. And you can maybe see here, so the cristae tips where it bends around are very curved, and the cristae junctions are very curved, and the cristae themselves are very flat. And something I always like to remind people is that membranes, if you had an infinite size membrane with no force on it, it would barely be curved at all. You would basically have like a sheet or a big circle. Membranes don't want to bend, they're rigid-ish. And so anywhere that you see membranes bending, you know that there's force being applied to the membrane, either by the accumulation of specific lipids or by proteins that bend the membrane. And so because of that, we can make a lot of inferences about where membrane bending force is being generated by creating these local maps of where curvature is in the tomogram. Finally, you can look at like orientation, and that seems like a very strange thing to look at in cells, but surprisingly, there are a lot of features in mitochondria, at least, that we know from high resolution light microscopy are very anisotropic in mitochondria. And so we know that, for instance, the proteins that are part of the mucose complex, which is what makes mitochondrial junctions, cristae junctions, are not equally found around the tube of the mitochondrion. They're on the sides only. And so we can really measure that distribution directly in the tomography data by looking at this orientation. And, it's great that we can do these local visualizations. We gain a lot from that. We can also aggregate that information and compare across many tomograms and then really have robust ways to do statistical analysis. So this is really the figure that convinces me that this is actually like a cell biology tool now, is that we can do good statistics using these segmentations and quantifications and do it with a lot of confidence. So just to give you a couple of examples, I talked a little bit about these arrows are pointing out the cristae junctions, but for surface distance, you can measure really effectively. You just take every triangle and measure the distance to every triangle on the other surface. We did this for both sort of the elongated and the fragmented population. We saw pretty big differences. We were quite surprised. You see sort of some regions in the fragmented that are quite far away. This sort of inner-outer membrane spacing is highly regulated, it turns out. We think that this is driving quite large-scale changes in function. And we can do a histogram. We can say, like, let's just look at the area of every triangle and add it into the bin of distance. There's lots of different ways you can sort of break down this measurement and take a look at it. So, we're making these measurements ultimately over millions of square nanometers of membrane, which is sort of the scale you're looking at in tomography, is the nanometer scale. And we can do it for one, but we can also, look at it for all of our tomograms, make that same measurement. And then by taking that measurement for each tomogram, we get this aggregation. And here I'm plotting the sort of mode distance between the inner and outer membrane. And what we saw is that the fragmented cells exhibit statistically significant increased OMM-IMM distance. You have like a 2.5 to 3.5 nanometer increase in spacing, but that the ER stress in the elongated cells actually decreased this distance by a statistically significant less than 1 nanometer difference. So we're sort of, we're in the range of the pixel sizes we're using. We're now sub-nanometer analysis, essentially. And, I think it's very important when you make these measurements to be like, is this something that's physiologically relevant? And we actually think that it is. Our collaborators have been studying this protein PSD1 that is a lipid processing protein that can act in trans across the membrane. And so this protein is, it's partially unstructured. But from our like coiled studies, this small decrease could actually lead to a 50% increase in trans interactions if it's acting just like a random coil reaching across. So we think that this PSD1 is able to act more in trans and convert phosphatidylserine to phosphatidylethanolamine much more efficiently, and sort of account for some of the loss of PE coming from the endoplasmic reticulum. So I mentioned we can also use this to like subcompartmentalize our features now. So this is something in mitochondria, but also many other organelles. Different parts of the organelle have their own functional characteristics. So the inner membrane of mitochondria is a great example of that, where the boundary membrane is where a lot of like protein import is organized. The cristae junctions are really highly regulatorily important because when they open up, that triggers the flooding out of cytochrome C, which is the first concerted step in the apoptosis machinery. So once the cytochrome C is out, this cell is getting ready to blow up. And then the cristae body, which is I've got in blue here, is where all of the energy generation is done. So the entire electron transport chain and ATP synthase and everything sits in these very sort of long, narrow cristae, which is where you can sort of generate a proton gradient that's why I describe it as capacitor. It basically is literally an electrical capacitor in addition to being a chemical capacitor that converts sort of your biological energy into ATP that the cells can use. And what we're doing here is we're just painting based on the distance from the outer membrane, these different features. And we can basically immediately segment out the junctions from the cristae bodies from the inner boundary membrane. This gives us the opportunity to really dig into what's different in those places. So we can look at the spacing within and between subcompartments and begin to make those measurements. So we can look at like intra-cristae spacing. So in the cristae bodies, what's the distance to the next crista over? Or I guess what's the distance to the other side of the crista, and then what's the distance to the next crista over? We see significant changes in intra-crista spacing, but it's surprisingly like the elongated, they tighten up, and the fragmented ones, they're ballooning. We think in the fragmented ones, we're actually seeing the beginning of that cytochrome C release that I was describing, where you're losing the regulation of the cristae junction and the whole thing is kind of coming open and is going to, if we watch these cells for 2 more hours instead of plunge freezing them, the fragmented population might have all died. You can also use these subcompartments for curvature, and this was really important because here, even though I'm showing you these 2 examples, looking at them, it's very tempting to say, well, they look really different in curvature. But when we did the measurement, the distribution of curvatures in the mitochondrial membrane was about the same. What's really happening is where the curvature has changed quite a lot. And so we've moved from having curvature that's sort of well distributed throughout the cristae to highly organized cristae with very highly curved cristae tips and cristae junctions. And we see this in the elongated population. The junctions are more curved and the cristae are less curved, and exactly the opposite thing is happening in the fragmented mitochondria. So we can do all of the statistics for this as well. And it wouldn't be cell biology if you didn't at least try to show that it's down the mechanistic pathway you think it is. So we've been doing co-treatments. So we think that this is a PERK pathway dependent effect. And here is the evidence is if we block PERK with a stress pathway inhibitor, the cells return to their original sort of healthier looking state. So basically, by taking advantage of this ultrastructural analysis, we were able to really dig into these aspects of cristae organization and mitochondrial remodeling at a scale that's really almost impossible to observe by anything except for cryo-electron tomography. And we were able to do it in such a way that we could really dig into, changes to the surface area, changes to control by mucose, and loss of regulation by ATP synthase. and changes to what we think is an off-sauce machinery. So, I was really excited about this. This was sort of the proof of principle of why this sort of ultrastructure first approach was great. But, I did mention at the front that, what we want to do is all three in one paper. You want to be doing not just ultrastructure, but how protein structure is impacting it and how these different scales are interacting. And that's not in this paper, really. But right now, I want to encourage you to be thinking about this as a platform for doing more than just the ultrastructural analysis. So there's a few ways people are doing this already, and there's going to be more in the future. But one is that you can use the surface to help you find proteins. So the sort of maybe dirty secret of subtomogram averaging that you'll hear more about later today is that the vast majority of subtomogram averages involve some amount of manual particle picking and some amount of prior orientation information. And so it's really hard to solve a structure. It's really hard to find your protein without kind of knowing it's near something that you know where that is. So in this case, I'm showing you can use the membrane and you can take your surface and you actually remap your structure, kind of like unrolling an orange. And you create it so that X and Y are now along the membrane, and then Z is stepping away from the membrane. And what's cool about that is you can now step up and you can be at a set distance from the membrane and see all of the membrane proteins at that distance from the membrane pop into view. And this is a really powerful way to sort of gain insight into where proteins are to make it easier to pick particles. And you get a lot of pre-orientation information because most proteins, they're sticking straight down into the membrane. So you actually gain all but the in-plane rotation angle just from the way you pick the particle. So here I'm showing you a proof of principle using rough ER. So this is an endoplasmic reticulum. It's called rough ER because it has ribosomes decorating it. And here I'm showing you two different patches that we've taken out and done this unrolling on. And you can see, hopefully, these black dots that are coming up all around the sides here. These are ribosomes that are attached. But now that you're looking at this way, hopefully it becomes clear, oh, they're forming this long line. They're not just randomly distributed on the membrane. And we think what you're seeing here is multiple ribosomes processing the same long piece of mRNA. So you're seeing translation of these membrane proteins by ribosomes into the ER membrane directly. But this works for smaller proteins. It works for a lot of different things. And, in my opinion, this is going to be one of the best ways forward for finding proteins that are sparsely decorating membranes. Membranorama was first developed by Dmitry Teganov and was in this 2020 paper. It's a Windows-only software. It's very manual. But more recently, a group including Kevin Yamauchi, Kyle Harrison, and Laurence Lam have developed this tool, Surforama, that is implementing a very similar approach in Napari. And so Napari is in Python land. It's very easy to build tools with it. And so now we're able to use sort of our surface tools in Python in the world that we feel comfortable in and do this kind of manual picking or just visualization in this really nice environment that's cross platforms. That's manual picking, but Laurence Lam, again, the same guy, has been working for a long time on ways to pick particles, not just manually, but automatically using your membrane surfaces. And so this is this tool, Membrane Pick. We're going to be talking about membrane seg later, which is a way to get your membrane segmentations quickly. And this is a way to automatically pick particles in membranes using a neural net model to identify things that are sort of bumping off of the membrane. If your particles aren't sparse, but instead are like super decorated on the membrane, a good example of that would be like a virus or, a chemosensory machinery. Instead of doing this kind of picking, you can actually do direct subtomogram averaging using the membrane mesh. So this is something that the idea has been around for a while, that if you build the mesh, you can oversample it. You can say, let's just pick every triangle on the mesh and average them together. We know their pre-orientation. And then you'll slowly get to the structure of the thing you're trying to find. And it turns out how a ton of different membrane structures have been found. I don't know exactly what you guys are going to be doing subtomogram averaging on later, but the HIV structure that was sort of the first dosimetric tomogram structure where people got really high resolution. People were drawing spheres, which is nice because viruses are spherical. It's much easier to make a model, and using it to do overpicking almost exactly like this. And so there's new tools like stopgap for doing that. I've been talking about membrane. I also wanted to mention that filament solving is also usually segmentation assisted. So this is something where, the majority of filament structures, people are segmenting the filaments, making models of them, and then using that to guide helical averaging or even non-helical averaging by picking particles all along the filament. And so this is, one of the papers that I think is the formative papers of modern cryo-electron tomography from Elizabeth Villa's lab, where they solved the structure of LRRK2, a protein whose structure was previously unknown, and they solved it within a cell first by identifying, decorating these microtubules and being able to take advantage of the microtubule geometry to solve the structure and identify this filamentous protein aggregate, or protein oligomer, I should say. Aggregate's a bad word in this case. I wanted to talk about one more thing before I get into the how part, because this is like what I'm doing right now. And it's always more fun for me to talk about like what's really exciting me right now, which is to use the membrane segmentation in the model as a sampling tool for digging into the mesh and digging into the density itself. So much in the same way that you can use an atomic model to ask questions about the EM density in its structure, you can use a mesh model for a membrane to ask questions about the density in the tomogram. And specifically, I started working on this to measure membrane thickness. So people have been doing this for a little while, but membranes have thickness that's very dependent on the type of lipids that are there. And they actually can form these things called lipid rafts in some cases that are where lipids accumulate and you get clearly visible differences in membrane thickness. And so these tools exist in single particle work where you're purifying these liposomes and you have perfectly circular membranes. But because we have our mesh that's able to represent a much more complex membrane, we can do the same kind of scanning in sort of biological membranes. And it turns out that in tomograms, low enough defocused tomograms, you have the leaflet information still there. So you can see the two leaflets. And so we can do our line scan. We can see if you just sort of scan through the normal, sort of perpendicular to the membrane, you can, much like quantifying a Western blot, for instance, you're doing essentially densitometry. You can measure this line scan, and then we can average them and get a really smooth curve. And we can see maybe in blue is the data. You can see maybe the data has these two bumps, and we can fit a pair of Gaussians to it. And use that to very sensitively measure the spacing between the two head groups of a lipid bilayer. And so we can do that for different components within a cell. And we did this in collaboration with someone that had identified in their condition like this crazy increase in thickness that they could even see by eye. And we were able to show we can see statistical significant differences even within a single tomogram between different features. And here the blue is ER and the red are these spherules that show up in these cells. And we see actually not only with our tool, we can see not only the difference between two different types of organelles, but we can see the range of results we see within these spherules. And we think that this is actually part of a maturation process. So you're sort of accumulating lipids and membrane proteins that are changing the local environment during that process. And so we're doing some sort of follow-up studies with our collaborators to try to dig into what exactly is happening there. But even not looking at weird cases, we can go back to our favorite organelle, the mitochondrion. And we can see this is a tomogram collected by Michaela Medina and Danielle Grochon's lab. That's the photograph there. The physiological organelle membranes have the same sort of amount of variance. So, we can see almost a 0.5 nanometer difference between the inner membrane and outer membrane of the mitochondrion. In this case, I like to think of I'm a protein structural biologist. When I see changes, I'm like, a protein's doing this. So some of this may be driven by differences in lipid composition, but really we think it's being driven by differences in proteins. The inner mitochondrial membrane is about 70% protein by mass. It's a very highly decorated membrane, and we think that it's being driven by is proteins that are deforming the lipid bilayer locally. And so, it's great to see this on the organelle level, but we kind of need to zoom in more. And we can do that. We can do local averaging instead of averaging over the whole organelle. And then what we can see is I've got this inset cut out because it can be a little bit complicated to look at these structures. Sorry, 1 second. And you see these red spots on this cristae. Those correspond to thicker sections. The blue sections are thinner. And we can zoom in on the red spots and go back to the tomogram in the same place. And what we see is they're being driven actually by these proteins that are in the membrane. These are electron transport chain components. And so now this is actually by far the most sensitive way that we've found so far to identify these proteins within our tomograms. And so it turns out that this local thickness measurement, which we were mostly doing out of curiosity and wanting to understand how the distribution of thickness happens within cells, is giving us a really powerful sort of handle to begin looking for membrane proteins that are very hard to find otherwise within cells. So, these are the kinds of things that I think in terms of sort of this ultrastructure down through structure, top-down approach to cellular biology is going to be really good for the field, hopefully, in the next few years. The last thing we're doing that I think is really hopefully cool to the structural biologists in the audience is, we're doing refinement now. So there's a lot of this idea that a model can only be as good as the data it's based on. And in this case, our meshes are based on the segmentation, and the segmentations are only so good. They're limited by quantization noise. But now that we're going back and directly sampling the data, we can get our models to be as good as the data. And we do that by saying, okay, we're measuring the spacing between our two head groups at each triangle, but we could actually also measure what the center is. And we notice that the center varies by several nanometers. We're not actually perfectly centering on the membrane at every triangle. We're doing an okay job. But instead here, what we can do is we can move every triangle in the direction that will put it right in the center. And so we can build meshes that are now a perfect representation of the lipid bilayer, or a more perfect representation of the lipid bilayer. And in doing so, we get a better mesh, we get better measurements. And you have this sort of iterative improvement that every time we make this, all of our measurements are less blurred out by this sort of local random variation. And we get sharper and sharper measurements in the same way that as you do refinement, you get better and better chemistry, better and better analysis of your protein structure. So, I think this is going to make all of the tools for, membrane-based averaging, surforama, and membranorama are going to work much better once we've taken our meshes, which were pretty good, and, made them much better. Because if you're moving things by, on average, maybe 2 nanometers in each direction, that's quite a bit for something like surforama, where you're stepping out by maybe, a few angstroms at a time, trying to find the plane where everything pops in together. So, this is stuff I'm really excited about. I think it's going to be hopefully, coming down the pipeline within the next few months. So as all of you that are learning about tomography now begin collecting your data, I hope that you'll keep in mind that these are the kinds of analyses that you can be doing with cellular tomography data. So I'm going to shift gears now and talk about not just why segmentation, but how segmentation. Because I think it can be a little bit of a dark art, even among tomographers. Every lab has their own sort of workflows for doing it, and many of them are really painful. So this is how I learned to do tomography. I thought I would show it to you because I want, like, it's very important to me to sort of emphasize that, like, right now is this incredibly exciting time where all of these things are getting way easier. So this is what I would do. I would go into iMod and I would use this live wire tool and I would just click along the membrane. Make sure that my thing is tracing. This is just the outer membrane. You'll notice there's quite a few membranes in this structure. And this is the simplest one. And then I'd hit OK. I'd, go down, scroll to another contour. And I would copy it in, and then I'd have to adjust it so that it was still tracing, because the membranes are not in the same place when you change your Z height. And I would do this. So it's sped up now, and it's sped up for a reason, which is just doing this membrane takes quite a while. And then I would have to mesh it. And for this one, it just works. For the really complex membrane, sometimes your meshing fails and you get to start over. And maybe you can imagine that doing this, you might spend 3-4 days on one tomogram. So if you're thinking about doing statistics and you're like, oh, I want to be able to solve, what's happening to 30, 50, 200 tomograms, you're looking at literally years of work potentially to be able to get all of these segmentations done. So I was very fortunate. I did this for 2 tomograms and was like, this is not sustainable. And thankfully, Ben Engel, who at the time was at BioZentrum, was willing to share their protocol for segmentation with me. And that's what we used for our paper. And this is using Amira. So I'm going to do sort of a more modernized version of this as a demo once we're done. I was hoping to go through the whole membrane seg pipeline. Unfortunately, my workstation that I was trying to do this on, membrane has been crashing for me consistently all morning. So I have it pre-computed and loaded into the structure. But this is TomoSegm MTV. You can see that the membranes are there and are nice, but they're also not very complete. There's lots of holes and there's lots of this kind of dust on the side. And so we're able to sort of magic wand and get our stuff really quickly, which is wonderful. You can really kind of, zip through everything compared to what we were doing before. But you do have to spend a lot of time like cleaning up, adding in stuff you missed, making sure that you have a reasonably complete segmentation. So the outer membrane goes quite quickly. The inner membrane is more complicated, and often it's more complicated than this even. But let's let it do its thing. This was like when we got this working, I made this video for my lab because it was me and 2 other people, including Michaela, who I mentioned earlier, collected some of this data, had been doing everything in iMod. And the video of me segmenting what we thought was going to take us like a week, because this has 3 mitochondria that are all complex, was this extreme moment of excitement for us. This software, TomoSegm MTV, is actually not new, but it's quite complex to use well. So Ben's protocol was really what enabled us to, very quickly be able to iterate through stuff. We get what's a pretty good segmentation in the end, but maybe you can see that there's lots of missing bits of membrane already where it doesn't do a perfect job. So what is TomoSegm MTV? It's a tool from Antonio Martinez Sanchez that involves computer vision, but not machine learning. It's using classical computer vision algorithms where there's a local edge detector or ridge detector, depending. And then the really special thing that they're doing, the reason it's called TomoSegm MTV, is that they do a tensor voting step where they sort of know that membranes should be roughly flat when they're right next to each other. They should have the same orientation. And so it's able to deal with a lot of the little bits of dust that you can see here by iteratively doing this processing. So this was a really powerful innovation. it works beautifully for these kinds of very simple spherical membranes. For the highly curved cellular membranes we were looking at, the problem is that the assumption that these two have to be not super curved is bad when sometimes you have really tight curvature. And so it fails often to recover high curvature. At the same time, we were using semi-automated filament segmentation in Amira. So filaments are the other side of this. I talked a lot about membranes, but that's another sort of ultrastructural feature we care about. And so the best tool still, and the tool I did a demo of last year, is this X tracing workflow where you do a cylinder correlation, and then you're able to basically use those cylinder correlations with pre-orientation information to trace through in an automated way, and pick out your filaments. The downside of filament segmentation, and I think something that's going to be fixed in the very near future, is that when you do this tracing, you end up with a huge number of false positives. So it runs and you get maybe 1,000 filaments, and only 50 to 100 of them are real. And so you have to go through and very carefully remove the bad ones. What I'm excited about that I'm not going to talk about today, but that my lab has just started doing, I don't feel very confident demoing it, which is why I'm not going to, because this is a thing where my grad students are now considerably better than me at it. And so there's a little bit of, I can't, I'm the wrong person to teach you too much about it, is Dragonfly, which is a commercial software that's free for academics, that has a really nice interface for building training data for doing neural network segmentation. So it makes it really easy to train. And with it, you can build neural networks that segment all kinds of different filaments in your cell. So the workflows for this have really been developed mostly in Matt Swulius's lab at Penn State. And they've been able to show that, for instance, their network is consistently able to differentiate actin from cofalactin, which is a pretty subtle difference and not one that I feel super confident in seeing by eye, even though an expert would be able to distinguish them. So with this, they've shown that if you do pre-segmentation with Dragonfly and then pass it into the cylinder tracing, you can get filament models for like subtomogram averaging and ultrastructural analysis without any need for like false positive removal, because they've already sort of cleaned up the search space. So finally, I just wanted to mention what we're going to be looking at today. I wanted to do a demo. I think it's a really important part of, when you learn about segmentation, it can be really hard to understand what it looks like to do it on the day-to-day, and you guys are taking this class to learn that. So this is another tool developed last year by Lawrence Lamb. I've mentioned him, I think, 3 times. He's working with Ben Engel and has been doing some of the really fundamental improvement work to doing segmentation better. So this tool, Membrane Seg, is a pre-trained neural network, so you don't have to do any of your own training. And it will do a much better job of segmenting membranes. You can probably see there's essentially no dust in this tomogram. So this gives us the opportunity to very quickly segment and get all of the features that we want out. And I'm going to show you a demo of that today. The last thing I wanted to highlight is, we're talking a lot about training neural networks. And a big part of that is it's a pain to prepare the training data for it. So the reason that Membrane Seg works so well is that Ben Engel is one of the best segmenters in the world and has hundreds to thousands of tomograms segmented that were able to be passed into this training data. But, we don't want to be doing that for everything. And increasingly, there are new tools coming out from multiple labs that enable simulation of cell-like data. And there's a lot of evidence so far indicating that you can sort of look at mixtures of the simulated data and the real trained data, and you can get like a 10-fold improvement over just looking at the human training on its own, because you can just get much more data very quickly this way. So I think in the not too distant future, the way that membrane has sort of, for us, solved membrane segmentation now, I think the same will become true for a lot of the other aspects of segmentation, including filaments and membrane proteins and other features. So I'm going to pause here. At my lab, I've gotten to work with a lot of different people that I wanted to acknowledge, particularly Michaela Medina and Eva Bonas, who I showed some of their stuff. My lab is very new. It opened about 5 months ago, so I feel I would be remiss if I didn't mention that I'm looking for postdocs. And if you're taking this course and you're like, you know what, I really love this, I want to do tomography and I want to do a lot of segmentation and think about cells, maybe give that a scan. And reach out if you'd like to talk, or just if you want to talk about your data and how to process it better. I'm very happy to do that. So I think now would probably be a good place to stop, take questions, maybe take a moment to breathe before we switch over to the demo, which I think is going to be less than 30 minutes. So you guys will have a little potentially coffee break, or we can have a discussion and you can ask me lots of questions. So, please, I love talking about this. I just spent a week in Michigan chatting with people about cryo-ET processing. So, is there anything anyone would like to ask?
Can I ask a question?
Of course.
Hi, it's a great talk. Thank you very much. And the question is, were you able to localize a protein fluorescence signal with your tomographs of the membranes to kind of localize to see where the proteins are and the static structures around it?
Yeah, that's a great question. So what you're describing is a technique called CLEM that is certainly possible. There's a big challenge with it. So those tomograms I was showing you, the field of view was 1 micron by 1 micron. And so when you're thinking about doing co-localization, your fluorescence signal with the typical fluorescence, you're getting sort of maybe a 200 nanometer radius spot, right? Your resolution is quite limited. So you can sort of find yourself to be, in the right quadrant of the tomogram. But it's very hard to do like, oh, this specific part of this membrane has these features. Some of this is limited by the way that we do cryo light microscopy. So in particular, we're limited to air objectives, which really makes a lot of the super resolution techniques quite difficult. I think that over time, this is going to get easier. And there are super resolution techniques like STED, where it would be possible to get much higher resolution. And then I think it will be very interesting to sort of go back once there are workable cryo-STED approaches or other cryo super resolution approaches to begin to do this, the kinds of fluorescence approaches that you're looking for. Because of that, what I'm really thinking about for this is instead I look to do perturbation. So I'm like, can I make this protein not go there anymore? Can I knock it out? Can I overexpress it? Can I add a drug that binds it, traffics it somewhere else? And then look at what changes in the tomogram. So that's a way to get at some of the molecular specificity magic of fluorescence work, but to do it without the sort of technical limitations of modern, not necessarily future, fluorescence light microscopy on cryo tomogram, like cryo lamellae.
Thank you.
Sure. Looks like there's a couple things in the chat. I can't see the chat while I'm in share screen, so
I can read them out loud.
Yeah, please.
So first question, for a segmentation image, is there image quality requirement for a good segmentation, or how to balance the bin number file and the image quality?
The bin number, is that what you asked?
Yes, the second part of the question is, or how to balance the bin number file and image quality.
Yeah, this is a good question. So, what it comes down to is like, how good does your sample need to be able to segment it? And binning is this idea that like tomograms are massive. And so we usually work with them binned, and we bin them to varying degrees. So most ultrastructural features are nanometer scale. like a membrane is 3.5 angstroms across, an actin is 7 nanometers across, a microtubule is 25 nanometers across. And because of that, we typically work with at least 4-fold binning. We bin down to 10 to 15 angstroms per pixel. And we lose basically nothing doing that from making the segmentation, and you get more contrast. In terms of how good the tomogram needs to be, the tomograms that I showed you that we had used in the paper were all collected. So I was a pandemic postdoc. We were supposed to get our energy filter in June of 2020, and you can maybe imagine that that didn't happen. So all of the data I showed was collected without an energy filter. And like when I look back at it now, it's pretty hard for me to look at, if I'm being completely honest. I'm not very proud of that data. And you can still get beautiful segmentations from it. The flip side is the better the tomogram, the less work it is to segment usually. So if you're trying to do a lot of them, it helps to have much better tomograms. But in terms of binning, it's very easy to bin down quite a bit and still get really nice tomograms and really nice segmentations.
Okay, the second question, can you tell us more about the inconsistency problems that you experienced with recentering when you talk about using the 2 bilayers?
Yeah, why it's not working. So this is like a really, now we're going to get into the weeds, but I have a triangle mesh and I make a measurement per face, and triangles are connected at points, and I make a measurement per face. And so my 2 faces might have different centering numbers. And so like the obvious thing would be, well, you just push each face towards the direction it wants to go. But the problem with doing that is that the face is defined by these 3 points, the vertices that are connected to multiple triangles. So they each have multiple things, and so they would go in different directions, and you would lose the connectivity of those vertices. And so I'm basically trying to figure out how to take these measurements and do like a really slow iterative stepping to get the vertices into the right place. So it's less inconsistent and more of just like sort of an engineering challenge to get things just right. One possibility might be to just redo all of the measurements on the vertices and just push the vertices, because those are much simpler. The reason to do the face-oriented measurements is it's much better for the ultrastructural analysis and for particle picking, because each face has an area. And so I can do like area weighting for statistics and say if it's covering a large area, that's more meaningful than one that's covering a small area. But yeah, this is like, technical nerd stuff, but it is it's fun. I did want to highlight, I almost forgot, I put up a very detailed tutorial of my software and of a few other softwares on my website for this course at Michigan. But it's just publicly available. I'm probably going to rename it to just tutorial at some point, but I wanted the students there to have something very obvious to go to. So if you're interested in like trying stuff on your own after the demo, this is up there, and I'll probably post it on Twitter, I would guess, later today. So, just something you could take a look at. Yeah, we have time. Does anyone else have more questions?
Yeah, so there's one more question.
Oh, yeah.
You mentioned that local thickness is a good way to identify lipid/protein content. Does data processing in such loci allow visualization of protein structure as well?
It doesn't give us the ability to directly visualize the individual protein structures. They're still really noisy. But if you can pick your proteins with the orientation of the mesh, and some of those proteins are really hard to pick, it becomes, much more straightforward to average them and then to do subtomogram averaging with those particle picks. So, not to tip our hands too much, but Michaela, the grad student that I was showing her tomograms for this, is literally working on doing this right now. It's using these picks to do subtomogram averaging of a bunch of different ETC components in an automated way. So I'll probably have a better answer for whether you can do it in like 6 to 8 months, maybe sooner. Cool. If everyone is satisfied, you can also, during the demo, great chatting time. There's a certain, like, it's a very meditative process doing segmentation, so I'm happy to talk while I'm doing it. Yeah, I'll stop share because I have to switch it to, let's see. Hopefully this is still working. Yes, it looks
There's
good.
actually one more question in the chat.
Oh yeah, please. Yeah.
So the question is it possible to create a grid and tile the region? Sorry, is it possible to create a grid and tile the region of interest as it is done in finite element analysis, FET, then process the nodes?
I don't know a lot about this kind of analysis. This is something that sounds interesting to me. My instinct is that like the proper regridding is reasonably possible, and there's a lot that can be improved in the way that we handle our triangle meshes. And if that's what you're asking about, I'm guessing the answer is like probably yes, but I haven't figured it out yet. Let's see. Okay. I'm remoted into my workstation. Can everyone see it? Hopefully, yes.
Yes.
This is Amira. Amira is a paid software from Thermo Fisher. It is sort of the standard in the field for looking at segmentation. But increasingly, people are switching to Dragonfly. People in my lab are using Dragonfly now. I am not very comfortable in Dragonfly, so I'm doing this in Amira. This is part of, getting to this point in my life, is that I now have my tools, and even though the field is changing, I am not. So I'm just scrolling up and down showing you this tomogram. It can be a little tricky over Zoom to see these live videos, but hopefully you can see there's a bunch of mitochondria in here. I can tighten up the contrast a little bit, and that might make it easier to see. There's mitochondria, there's ribosomes, there's things you can actually see in some cases the bilayer. If I zoom in.
Excuse me, can I just ask an off-topic question?
Always, I love off-topic questions.
Yeah, because it's about the mitochondria, not necessarily cryo-ET. So
Okay.
yeah, looks like you are using mitochondria a lot, or you are looking at the mitochondria a lot, right?
Yeah.
Yeah, I'm curious, have you ever seen ER rings surrounding mitochondria contract?
You're asking about like the fission ring model?
Yeah, have you ever seen the DRP1 rings?
We haven't seen DRP1 rings. We've seen things we think might be, but they're not there at most of our CLEM sites, including DRP1 CLEM.
Okay, good to know.
With that said, like we think that it's probably because it's very transient, right? Like it forms sort of because DRP1 comes on and off the membrane all the time, and it only forms the ring at the very end. And so our suspicion is that we are just, the downside of being a snapshot technique is that if your snapshot is not at the exact right time, you're not going to get it. So this is an active area of research for multiple labs right now. I would guess that, progress will be made on that. The first question you asked was about ER encircling, and that we definitely do see ER crossing over, but we rarely see like the complete rings like what the some of the models have depicted. Instead, I think that there's clearly ER contact sites, but it's a little bit more complex. We published a paper last year. I'm like the middleist author on it because I did some filament quantification, and that was all I did for it. It was showing some of these sort of features that we saw in the data. And I'd encourage you to take a look at that. It's Magda Sweron et al. And it's Grant Jensen is the last author.
Okay, I saw that paper.
Yeah, awesome. I'm happy to hear it.
Thanks.
So we've seen this, and now I want to show you what membrane looks like. And I wanted to let you know this membrane structure was generated in about 5 minutes with no cleanup prior to this. So rather than having huge amounts of noise, now we're able to really, there we go, really scan back up and down and see almost perfectly just our features of interest are here. And I can even, I'm going to show you, we've already, let's do voxelized rendering. And now you can see this is the completed segmentation. I don't think I'm gonna have time to complete the whole thing, but you can see it basically snaps into this beautiful structure, and there's only a few places where the membranes that were segmented aren't real. So it's possible to now generate these really, I think, beautiful, let's put that away and do this. This is what I wanted. These beautiful segmentations in very little time. And this is enabling people to think now about not just doing segmentation in a small number of tomograms, but looking at 80, 100, 200 tomograms. And that's really where you start to get some power. So I'm going to switch us over to a new segmentation. And I'm just going to show you, like, once you have a pre-seg like this, just how convenient this process can be. So we're going to move this over. I am going to just show the XY view. Normally, I would use multiple panels so I could see the 3D as I go, but I just want things to be simple. So we're going to start by segmenting the IMM. And we have this tool that is called the magic wand. You can tell it to just select all slices. Okay. Let's see. There we go. Okay. And select bits. So there's something happening here, which is that we're bridging these two. And so we can adjust our masking a little bit. Yeah. And so I'm going to just add these to the IMM for the moment. Yeah. Selection add. Change the color of this so it's very different looking. Add. So now if I switch to the, I'm going to turn on the 3D. If I switch back here, you can see we segmented these two membranes, but they're bridged. So it's segmenting the inner and outer membrane as one. And so we're going to fix that at the end, but for now, I'm just going to go forward and keep going. So, as far as workflows, this is pretty painless. So this is inner membrane. Oh. Switch us back to the single pane view. And we can change the display if we ever need to. We can say, I want to see the tomogram. Actually, let me do it this way. We're just going to switch back over to project view. Hide this, show this, and just make sure the features that I'm thinking about segmenting are mitochondria. It's always a good idea to not segment too blind.
Excuse me, another question.
Hit me.
Yeah, so like this type of work, including cryofiber sample prep, and
Yeah.
image processing, then segmentation. Which part do you think is most time consuming?
Segmentation.
Segmentation, really?
It's changing, but yeah, I mean, it's very, it's a very slow process for sure.
Compared to like a cryo-fiber milling?
Well, the fib milling, what's really cool is how automated it's become. It's definitely very time intensive, but like, you know, I typically, the last data set I milled, which was in probably October, because I'm starting a new lab and that means I don't get to do fun stuff for a moment. This is my fun activity now, is I am able to mill maybe 30 lamellae
A
in
day,
an overnight session.
overnight.
And that's fast, but it's because I make smaller lamellae. And then on the cryos from those 30 lamellae, I might be able to collect in a day 150 tomograms. And so, you know, 2 days of work and you're now it's hard because if you have to do it right, right? The nice thing about segmentation is it's not really hard. You can just kind of do it. It takes a long time, but you get a good result, even if you don't know what you're doing, which I don't half the time. Versus if you know what you're doing, the cryos and the data collection can go quite fast now. And this is a recent change. There's been about a 5-fold speed up in everything since 2019 because of advances to the way we do beam shift, image shift data collection, and the way we automate the milling. But with that said, because of that, the heavy load is very much on the data processing now. It's aligning tilt series well. There's a lot of manual steps in that. It's particle picking. It's segmentation. That is very much sort of the challenge of tomography is that, you know, segmentation is there's a lot of steps that are not fully automated yet,
right?
And that we'd like to be automated.
Okay, you mentioned that the data acquisition. So what software are you using? How you speed it up?
So 2 years ago, there was a paper called Pacetomo from Fabian Eisenstein in Rado Donev's lab. And what they did was they did all of this amazing math that let them do beam shift, image shift with tilting. So they predict when you tilt a sample where the stuff is going to move so they can beam shift, image shift to the new thing. And it allows you to take many images per tilt series now.
It's in series,
and it means
right?
that we typically spend about 4 minutes on each tilt series instead of we used to spend 30.
That's great.
And that's implemented in serial-EM,
Serial.
using the Python interface in serial-EM. But since then, because it works so well and because, you know, Fabian did a really good job describing exactly the math to do it. It wasn't new math. It was actually math from a 2000s David Agard paper. Thermo Fisher has also implemented the same beam shift, image shift now in Tomo 5. So you do very similar workflows now in both softwares. And in doing so, you're really able to get access to a lot of data very quickly.
Great.
Yeah. You know, the process is not trivial yet, but it's gotten much, much easier to do.
So what you use iMod for processing?
So I can tell you, it might be interesting to talk about sort of my data processing stack in my lab. So we do all of our pre-processing on the fly with Warp, and then generate motion corrected CTF corrected tilt series. We then do alignment with iMod, and we do batch alignment, but it's manual. We're not doing any R-tomo. What we found is that if you spend the time to very carefully align each tomogram better, it makes the downstream processing faster enough to make up for that cost. Everything is easier basically with a better aligned tomogram. We then do reconstruction with Warp. and make half maps and then do denoising with cryo-care, which is a denoising algorithm. We then sometimes will also pass it through ISONet to do missing wedge correction. And then from there, it'll go into membrane seg, which is different from membrane pick. And that's the segmentation that I'm sort of painting in right now.
Right.
As far as subtomogram averaging, we usually do, we've been doing PyTom particle picking, which is a template matching algorithm. But I don't know that I have a strong instinct that that is the best segmentation algorithm. It's just, you know, one that works.
Great. Thanks for
That,
sharing.
sure. Well, I think I underestimated how quick I would be at this. It's very funny because like it's been long enough since it's been me doing it that I'm now finding that I'm quite slow. But I will show you what we've done so far, and sort of we've grabbed the 3D mitochondria. We have a problem with, and this happens sometimes with membrane seg data, which is that there's crossovers, so the two sides get linked. And you basically, what you do is you have to scroll up and down in space until you find the point where they're connected. And you just have to remove that from the segmentation manually, and then you can snap them into their own separate classes. I had a really hard time finding it in this one, which is why I didn't just clean it up in front of you just now. Oh, here. So see that crossover right there? So we can switch to the brush. Make it smaller. Nice. Do that, and then I'm gonna actually mag in on this a little bit, make it easier to see what I'm doing. Oh, what is the one? And then you can step down through a few slices. And 2, 3, 4. And I'm doing this in a few places, and we're going to try to interpolate our way out of it. We're going to see if it works. So I'm going to hide these. That's it. Do it. And oh, sorry everyone. This is a live demo experience. I'm blocked by the screen share. Forget it. We'll just do Control-I. We should be able to scale through, and now you can see it's put them everywhere. Didn't do a great job, but it did a pretty good job. And we can then come to IMM and remove this to clean these up. And so we'll then iteratively go through and like finish removing these, which I'm not going to do. And then you can magic wand on same material only and switch the outer membrane. to outer membrane. So we're just about out of time. I want to keep you guys on time. I'm going to switch us over and just like very quickly show you what someone with lots of time that's doing a good job can do. Better than I can. So here is where these have been separated successfully. Got Golgi and nucleus, and you can see here we've got lots of different kinds of classes that have been separated out. We've got these multivesicular bodies that I grabbed by accident, the wrong thing. We have ER. You were asking about whether we see ER crossing over the mitochondrion. Here's a case where we're getting one of these crossover events. And you can get these very quick segmentations. Now it's like, we end up spending about 20 minutes to half an hour per tomogram when you're sitting and working really efficiently, which I was sort of doing an okay job today. And so, if you're thinking about segmenting hundreds of tomograms, it becomes more of like it's a couple of weeks of work to do that now. And it's to the point where you can really start thinking about doing this at large scale. This has been really fun. Thank you for inviting me out again to do this talk. I really enjoy presenting this to people that are curious about tomography and about electron microscopy. And I'm really hopeful that this becomes a field that's very easy to enter. It's become much easier to enter, but it can still be a little bit of an intimidating thing to start doing. And hopefully, even if I didn't really succeed in showing you the segmentation going super smoothly, I made it a little bit less scary looking at the point I was just driving around and painting. It's more of a sort of Bob Rossi experience. And yeah, are there any last questions before I hand it over? If not, thank you guys so much. I got lots of good questions during, and I really appreciate that very much.
Thank you, Ben. Thank you. It's always a pleasure. It's an excellent talk and presentation. It's very exciting.